Welcome back to Module 4. This tool is called pSORTb. Remember, this whole module is about cellular localization data. So it's predicting where this protein of yours from this gene of yours is residing in the cell, or if it's meant to be secreted out of the cell, as in the case of, say, a toxin, where the bacteria oozes it out of itself to poison other things nearby it or to help it compete, or if it is something that embeds in the membrane with a transmembrane helix. So this tool is pSORTb. I'm in my notebook for KSED00010, and you control click to get there, and you can see that we're set up here. We have bacterial genes, so your Clostridium, you want that to say bacteria. The gram stain for Clostridium botulinum you need to select. For KSED00010, we just discovered in this lipo-P section that it's gram positive. So we would want to select that, and now we need our FASTA sequence. So we go back to our basic info and make sure we grab the FASTA header and the nucleotide sequence. We want the amino acid sequence. Don't get anything extra. Go back to pSORTb, paste it in. The last thing you want to change is to make sure that you have via the web, or else it's going to make you email, and it just doesn't take that long. So do via the web. And if you ever have to go back, this is something that you have to have all the required things done. Okay, and select via the web. So submit it. And I'm in the manual where it says record all the data into the notebook, the numbers, the for cytoplasmic membrane, cell wall, extracellular scores. And it says in bold, this is just page 92. If any one score is greater than 7.5, it will be the final predicted location for your protein. If no score is greater than 7.5, the prediction may be that the protein can exist in multiple sites. If all scores are equal, then pSORTb is unable to assign a final predicted location. So this is doing the same thing as our TMHMM. Okay, so if I go back to the KSED results, you can see that it says with this localization scores, cytoplasmic 9.97. So that is a really high score, so its final prediction is that it's cytoplasmic. So if I go to GENI-ACT again, I go back to pSORTb, my cytoplasmic score, all I have to do is get it, 9.97, and save. Cytoplasmic membrane score, 0.00. Save it. Cell wall score, 0.01. Save it. Periplasmic score, which shouldn't apply, so it doesn't even list it. A periplasmic score would be if your bacteria is gram negative. So if Clostridium is gram negative, you would see a periplasmic score. But there isn't one. So I'm not going to leave a blank box. I'm going to say not applicable again, so that I don't get freaked out if I see a blank one later. Same thing, there isn't an outer membrane, so you could see there's not an outer membrane option. Chytococcus is gram positive. It doesn't have an outer membrane or a periplasmic space. Extracellular though, well, that means it's secreted or something, maybe 0.02. Save it. pSORT final prediction. So I said it was cytoplasmic. It had a 9.97 score, but I'm kind of reminding myself from the manual that 7.5 is really significant. And I save it, and that's it for pSORTb.